appreciate it, and then move on. Okay, so this week is integrated meal. I wanted to start off by saying all these 3 units for this exam are very closely intertwined. So when you're studying, I suggest kind of looking at them all as one, like big picture, kind of going through, and looking at like the details from the first unit, and then also this unit, how the food is kind of like processed in each system. And if you look at my study guide, which I put at the end of this, that's how I went about it too. Just kind of like looking at everything cohesively, I think is really helpful. So again, my office hours are tomorrow, 2:30 to 3:30 in row. There's my email. And I mentioned this last week, or last SI, that I don't have a typed study guide for this unit, but I have a written study guide available at the end of these slides. So I'm sorry about that, but I don't know why I didn't type this unit, but I have it written. And then I also have the practical study guides on Husky CT, if you're starting to prepare for the next practical, which I don't think is for a while, but just a reminder. Okay, so I wanted to start integrated meal by kind of zooming out and talking about how the GI tract is just one long tube through your body that the food kind of gets processed by mechanically and chemically to absorb all the nutrients. So we talked about this tract last SI session, so that video is available on Husky CT. And then remember that movement between different organs of the GI system is controlled by the sphincters. And we talked about how these can be voluntary or involuntary based off of the type of muscle that they're made of. And then each different region has different secretory and motor behaviors to digest the food. And it's separated into 4 different main phases, which are the cephalic phase, gastric phase, intestinal, and colonic phase. So I'm going to go through each of those in the coming slides. So the cephalic phase is digestion in the head and esophagus, so it's like the food enters your mouth, goes down the esophagus, and then you have the upper esophageal sphincter and the lower esophageal sphincter, which we'll talk about soon in a few slides. So chemical digestion begins right in the mouth, where you have your saliva, which contains digestive enzymes. So you have salivary amylase and salivary lipase in the saliva itself. Amylase starts to digest starch into disaccharides right away. So starch is just like big carbohydrates get digested right down into 2-part carbohydrates in the mouth. And then salivary lipase starts to work on fats right away. Note that there is nothing here digesting proteins. And then you have some protective enzymes in saliva, including fluoride, lysozyme, and bicarbonate. So that just helps protect your body from any pathogens that might be on the food that you're eating or that enter your mouth. So then obviously you swallow to move your food from your mouth into the esophagus, and it's controlled by these 2 sphincters. So you have the upper esophageal sphincter. This is skeletal muscle, which tells you that it's voluntary, and that's how you can control when you swallow. The lower esophageal sphincter is at the bottom of your esophagus, between the esophagus and the stomach. And this one is made of smooth muscle, which tells you that it's not voluntary, or it's autonomic. and this one opens when the pressure in the esophagus is greater than the pressure in the stomach. Okay, so I was gonna draw this, but I think that I'll save the drawing for more complex concepts later. But I just wanted to emphasize where the upper esophageal sphincter is between like your mouth and your esophagus, and how it is voluntary and made of skeletal muscle, and then how the lower one is involuntary. And then I also wanted to mention gastroesophageal reflux. I think this is kind of cool because it makes sense. It's caused when the pressure in the esophagus is less than the pressure in the stomach, which causes that sphincter to just open and allow food back up into the esophagus because of that pressure difference. So there are some foods that naturally kind of reduce the pressure in the esophagus, which promotes like acid reflux, which include caffeine, alcohol, cigarettes, and chocolate. Any questions so far? Now moving into phase 2, the gastric phase. This is your stomach. Function here is digestion by secreting enzymes from the gastric pits, and it's not really for absorption. It's not a main function of the stomach. And then 2 ways to divide the stomach. You have the anatomical divisions and the functional divisions. Talking about the functional divisions, you have the proximal and distal stomach. The proximal stomach is for storage, and it has specially like rugae for that. And the distal stomach has an extra layer of muscle that helps kind of churn the mixture of food in there to help it digest mechanically. So here's a chart. I think I took this from one of the lecture videos that shows the secretions in the stomach. And something to note is that most of the digestive secretions are in the fundus and body. So that's where you have a lot of these gastric pit cells that do all of the secretions and help with digestion in the stomach. So you have chief cells that secrete pepsinogen, and pepsinogen is converted to pepsin in the presence of acid, which then helps digest proteins. Parietal cells secrete HCl, an intrinsic factor. Mucus cells produce mucus. G cells produce gastrin. D cells somatostatin, and ECL cells histamine. So going further into protection of the stomach, so those mucus cells are really important to kind of produce a mucus layer on the stomach. Since the pH of the stomach is really acidic, it's pH of 2. And then there's also like pepsinogen getting converted to pepsin, which digests proteins. So the mucus cells are really important to produce that barrier, because without it, then pepsin would just start digesting like the wall of the stomach. So when that does happen, you have some gastric ulcers that are formed. And it's also important that pepsin is only activated into pepsin. No, that's the reverse. Pepsinogen is only activated to pepsin when food is present. On to phase 3, the small intestine. This is where a lot of absorption is taking place. But before we get into the small intestine, we have to empty from the stomach into the small intestine. So gastric emptying is a highly regulated process. It's increased by a lot of food and protein in the stomach. So if you think about that logically, that makes sense. The stomach has a lot of food and protein, so then that promotes the food from being moved from the stomach to the small intestine. And then there's also neuronal control on the pyloric sphincter in between the stomach and the small intestine that helps promote like opening of the sphincter and moving food from the stomach to the small intestine. However, we don't want food to move from the stomach to the small intestine really fast, because then we have a lot of food in the small intestine that can't all get absorbed nicely. So enterogastrones are hormones that help decrease the movement from the stomach to the small intestine, so that we kind of digest slowly and can absorb all the nutrients from our food correctly. So these hormones are released from the small intestine, and they're triggered by food being in the duodenum, which is just the first region of the small intestine. So these three hormones are secretin, cholecystokinin, and gastric inhibitory peptide. And now we're going to learn more about those. So each hormone is released based off of a certain nutrient or a certain condition in the duodenum of the small intestine. Secretin is stimulated by acidic chyme entering the duodenum. So the chyme enters the duodenum from the stomach, secretin is released, which then inhibits the stomach from releasing more food. Basically just slows it down. At the same time, secretin reduces HCl production in the stomach. And then it also stimulates the pancreas to secrete bicarbonate into the lumen to neutralize that acid. So you get acid into the small intestine. You then want to inhibit gastric emptying to slow it down, reduce acid production in the stomach so we get less acid in the small intestine, coming into the small intestine, and secrete a basic compound to neutralize that acid in the small intestine. So if you think about it that way, it all kind of makes sense if you think it through. CCK, or cholecystokinin, is stimulated by fat entering the small intestine. So if there's fat in the food that you were eating and then it gets moved into the small intestine, CCK is released, and it inhibits gastric emptying. It just slows it down, and it inhibits gastric activity of pancreatic enzymes. GIP is instead stimulated or released due to sugar or glucose in the small intestine. And what it does is it stimulates the uptake and storage of glucose. So it's kind of a signal. It's like we have glucose in the small intestine, GIP is released, and it stimulates insulin release, kind of tells the body we have glucose coming in. So then right there we already have glucose or insulin getting released. And then insulin, just a reminder, is that hormone that helps take glucose into the cells to be used. So if insulin is the hormone that is released when we have high blood glucose. Does anyone remember the hormone that's released when we have low blood glucose? Have you talked about that yet? Or that might be the next unit, but that's glucagon, if you haven't talked about that yet. Yeah, we'll get into that later. So then dumping syndrome describes when gastric emptying is just happening too fast. So the enterogastrone hormones aren't working properly, and food is just leaving the stomach too quickly, which then drives water into the small intestine because you have a bunch of things that aren't getting absorbed into the blood, and when you have high solute concentrations, you're driving more water into the small intestine. So then that can lead to like diarrhea because you have a lot of water and your food isn't getting absorbed. And then to quickly just look at small intestine anatomy, zooming out, the duodenum is the region that where a lot of like digestive enzymes get released into. So there's a lot of digestion and mixing in this region. Jejunum has some more digestion, and then also is important for absorption. And then once you get down to the ileum, it's mostly absorption. Getting further into that first region of the small intestine, the duodenum, you have a lot of accessory organs releasing enzymes into this region to help complete chemical digestion. Some important ones are pancreatic amylase and lipase come from the pancreas, and then bile from the liver. Taking a side step into the accessory organs, the three accessory organs are the pancreas, liver, and gallbladder. The liver, main thing to know is that it produces bile. Gallbladder stores bile. And then the pancreas has two regions. First is the exocrine region, which is also known as the pancreatic acini. So if you look at that picture that I put on the slide, the acini are like these ducts right here, and they produce bicarbonate. Or the duct region produces the bicarbonate, and then the acinus produces pancreatic amylase, pancreatic lipase, and then zymogens, which are precursors to those two enzymes. And then relating this back to the duodenum, those two enzymes get released into the duodenum and help further digest carbs and fats to more simpler forms that could be absorbed into the wall of the small intestine later on in the jejunum or ileum of the small intestine. And then this relates to what we talked about last week if you watched the SI session. The endocrine region of the pancreas is known as the pancreatic islets, I think. And that's these like little blue flower looking structures on the diagram. So this is where the alpha and beta cells are. Alpha cells release glucagon, and the beta cells release insulin. So insulin is released in response to high blood glucose. Glucagon is released in response to low blood glucose. And if you go back and look at the slides from SI last week, we talked about how insulin is regulated by high blood glucose. But that I think you do talk about a lot more next week. So the slides that I made from last SI might be very helpful when you're going over the material for next week. Okay, I think this is where I want to see the drawing. Okay, yeah, we can draw this. Okay, so in this unit, you talk about kind of 3 main different nutrients, and especially in integrated meal, you're talking about how they're getting digested and absorbed in each region of the body or of the GI tract. So if we go through each nutrient, we can talk about how each enzyme kind of progresses digestion throughout the GI tract. So starting with carbs, no, sorry. And then the main regions were digestion. Now, lumen of the small intestine. So this is like the duodenum that we were just talking about. And then so these are just kind of main regions where digestion occurs. So does anyone remember where carbohydrate digestion starts? Which region?
No.
Yeah. So right away when carbs enter your mouth, chemical digestion starts right away. So carbs enter as polysaccharides. I'm just going to abbreviate that. And then in the mouth, salivary amylase. In the saliva, right away, breaks them down into disaccharides, or 2-part sugars. And then we move into the stomach, and there are no enzymes in the stomach that help with carbohydrate digestion. So we can just leave that blank. And then once we get to the lumen of the small intestine, we just talked about that pancreatic amylase from the pancreas. So more polysaccharides that weren't digested into disaccharides in the mouth are digested into disaccharides in the lumen of the small intestine. So again, we have more polysaccharides, and then are digested by the pancreatic amylase. Into disaccharides. And then the brush border refers to enzymes like in the intestinal epithelial cells. So lumen of the small intestine just refers to enzymes in the space of the duodenum in the small intestine. Now we're kind of, the brush border is more, since they're like villi, we're here now. So this is the lumen and this is brush border. So to get absorbed into the blood, we still have to get broken down into, remember, those simple sugars or monosaccharides. So in the brush border, we have disaccharidases, which are enzymes that break down disaccharides into monosaccharides. Monosaccharides. Does anyone remember what the three monosaccharides are?
Glucose, lactose, and fructose.
Perfect. I would definitely know those. So it's glucose, galactose, and fructose. And then the three disaccharides are maltose, sucrose, and lactose. And then once we're at this simple sugar level, we can get absorbed through the small intestine wall into the blood. And that's the goal. Switch colors. Does that help like break it down with like where all the enzymes are used? Okay. And then we'll go through protein. So in saliva there's only salivary amylase and lipase, so no protein digestion starts in the mouth. So we're going right into the stomach. And in the stomach, we're starting with protein. And then pepsin breaks proteins down into peptides, or just smaller parts of protein. And then we get into the small intestine, and pepsin is broken down by trypsin and chymotrypsin into smaller peptides. So now we're in the small intestine. And then there's another enzyme in the small intestine called carboxypeptidase that breaks down the smaller peptides into free amino acids. So we're still in the lumen of the small intestine. Carboxypeptidase. Into amino acids. of the smaller peptides that aren't broken down into amino acids in the lumen of the small intestine reach the brush border. And so we also have two more enzymes, aminopeptidase and dipeptidase, that make sure that all the proteins are broken down into amino acids so they can be absorbed into the blood. So smaller peptides that make it to the brush border. There's aminopeptidase. Which helps convert more to amino acids. And then in the case that the smaller peptide is a dipeptide, or just two peptides, you have dipeptidase, which then makes sure that we're in the amino acid form, so then we can be absorbed into the brush border and into the blood. And then the fat is pretty simple. There's no digestion until we get to the lumen of the small intestine. And that's where we have lipase. So fat, or also known as just triglycerides. And lipase is the enzyme that breaks fats down into fatty acids, which is just the monomer for monoglycerides. Something special about fat is these two, the goal was to get into the brush border, to get into the blood. Fat can dissolve, or not dissolve, can be transported right across the membrane since it's lipid soluble, and then it goes into the lymphatic system, or the lacteals, instead of blood vessels. But I have this on this chart, if it's more clear for you to reference it that way. It can just get kind of overwhelming with all the enzymes, so I feel like looking at it all in one piece of paper is helpful. So you can see, maybe even when you're studying, you can like circle the enzymes, so you can just remember where each one's used and what it breaks down. So if you did watch the video from last week, this is basically a lot of what we went over. And what this is describing is when we got to the simple sugars in carbohydrate digestion. This diagram shows how those simple sugars are absorbed into this intestinal epithelial cell and then into the blood. So there are those two transporters on the apical membrane here that allow for the sugars to go into the intestinal epithelial cell, and then that one transporter, GLUT2, that allows the sugars into the blood. Something to remember when talking about absorption is solubility is really important. So carbohydrates and proteins are polar, so they can't be transported or diffused right through the membrane. They need some sort of transporter. So on this slide, this is talking about carbohydrate absorption, we're using transporters to get across both membranes of the epithelial cell. Same thing with protein. You need a transporter whenever you're crossing a membrane with protein. Lipids, on the other hand, are fat soluble, so they can diffuse right through the membrane and don't need a transporter. But like you guys talked about last lecture, lipids kind of congregate into the micelles and are then absorbed into the membrane. Okay, so I was going to draw this quickly. So, again, in the duodenum of or just in the small intestine in general, those intestinal cells, so we're going to be at one of those intestinal cells. This is the apical membrane, so then this is the lumen of the small intestine. And you have a bunch of transporters here. And then this is the basolateral membrane. So then this is the blood. Like we talked about last week, there's 2 carbohydrate transporters. SGLT1 and GLUT5. Do these sound familiar? Did you guys go over these? Okay, good. So to review those, we talked about them last week, but SGLT1 is a symporter which transports glucose and galactose simple sugars into the intestinal epithelial cell via secondary active transport. So it allows sodium to diffuse down its concentration gradient into the intestinal cell, which powers glucose and galactose to be able to enter the intestinal epithelial cell. GLUT5 is for facilitated diffusion of fructose. And then once the carbs are in, or the simple sugars are in the intestinal epithelial cell, they then are going to want to enter the blood. So then there's another transporter on the basolateral membrane, which is GLUT2. And this one allows all the simple sugars to diffuse into the blood via facilitated diffusion. So they're at a high concentration gradient to a lower concentration gradient. So glucose, galactose, and fructose. Does that make sense for carbs? Okay. And now to this diagram, we're going to add protein absorption. So you have 2 protein transporters on the apical membrane. The first one is the amino acid sodium symporter. So I'm just going to write amino acid sodium symporter. So it's a symporter, which means it's transporting 2 things in one direction. so it's allowing sodium in and amino acids into the intestinal epithelial cell. And then the second one is the di or tripeptide hydrogen importer. Tripeptide. So now we're allowing hydrogen ions in and di/tripeptides in. So there's something a little bit weird about that because the amino acids are not all the way broken down and they're in the intestinal epithelial cell. So actually in the intestinal epithelial cell, there are enzymes that break down these di and tripeptides into amino acids. So then they can go through this transporter into the blood for proteins, which I don't think you have to know the name of, you just have to know that there's a transporter there that allows proteins into the blood via facilitated diffusion. So this is just a transporter. And the enzymes that break these di/tripeptides into amino acids are these ones right here, aminopeptidase and dipeptidase. At the brush border. So amino and dipeptidase. And then they're amino acids and can go into the blood. So that is more clear there. Notice how I didn't add fat to this diagram, again, because there are no transporters for fat. These fatty acids congregate into the micelle, which then gets absorbed into the same spot. And then I think it's the what is it? Yeah. They congregate into that and then get absorbed into the lacteal. Any questions on this at all? Okay, cool. Let's see. I just have a few more slides and then we can do a few top hat questions. So to help the movement through the small intestine, there's peristalsis, which just kind of the waves that kind of help push the food through. And then there's motilin, which is a hormone that's released during fasting to help stimulate strong peristaltic waves, which initiate a migrating motor complex to push any food that you have left through to try to like help you absorb as much nutrients as you can from any food remaining in the small intestine. And then the last phase is the colonic phase, which is the large intestine. Here are just the regions, cecum, colon, and rectum. And then I emphasized this last week too. There are two sphincters the internal anal sphincter and the external anal sphincter. So the internal one is smooth muscle, which helps you know that it's involuntary, whereas the external anal sphincter is skeletal muscle, which implies that it's voluntary. So skeletal muscle is typically something you can control, whereas smooth muscle is usually like an autonomic thing that's regulated more by pressure gradients or. Okay, let's see if this works. Okay. The lower esophageal sphincter opens to allow food into the stomach when the pressure in the stomach is greater than the pressure in the esophagus. True or false? Nice. So it's the opposite. The lower esophageal sphincter opens when pressure in the esophagus is greater than pressure in the stomach. This case would be like acid reflux if you have greater pressure in the stomach, causing the lower sphincter to open. Enterogastrones stimulate gastric emptying by releasing the enzymes CCK, GIP, and secretin. So this one, I'd maybe think back to that slide where I had stimulates gastric emptying and inhibits gastric emptying. If you remember, we talked about how the pyloric sphincter is innervated by nervous control, which can help stimulate gastric emptying, and also food in the stomach helps stimulate gastric emptying. Enterogastrones inhibit gastric emptying. Does that make sense? Okay. And then the enterogastrones are stimulated by something specific entering the duodenum, so just try to match which each one is stimulated by. Okay, seems like you guys did pretty good. Secretin is stimulated by acid, CCK, fat, GIP, glucose. Chemical digestion is completed in the stomach. True or false? So this one is false. If you look at this diagram that we drew, the only nutrient that's digested at all in the stomach is protein. Fat and protein and carbs continue chemical digestion in the small intestine, in the lumen of the duodenum, and then also at the brush border, with everything here down. So pancreatic amylase, trypsin, chymotrypsin. CCK is release is triggered by blink and leads to blink. Perfect. So CCK is released by, or is triggered by fats entering the duodenum, and it leads to bile emulsification by helping get those pancreatic lipase enzymes. Select the following nutrients that are not soluble in the membrane and therefore need a transporter to move across cells. Nice, yeah. So I just wanted to emphasize that proteins and carbs need these transporters, fats do not. Okay, so this one you guys have not done yet, I believe. This is next week. So I think I might actually skip this. You haven't heard of the arcuate nucleus, right? Okay, yeah, we'll do that next week. In what form are lipids absorbed into the small intestine cell, and then what form are they absorbed into lacteals? So I could honestly add this to this diagram. The lipids aggregate into the micelle, which then goes right through the membrane and then forms the chylomicron. Which then goes into the lacteals. And last one. You just had a meal rich in carbohydrates. What hormone is directly released due to an increased concentration of carbohydrates in circulation? Perfect. Insulin, and that's like a good segue into metabolism with next week. Yeah. Thank you guys for coming.